The enzyme superoxide dismutase is one of the body's foremost antioxidant defenses, reducing free radical damage that's associated with atherosclerosis, stroke, and arthritis. New bioavailable forms of superoxide dismutase offer superior protection against oxygen-free radicals. For years, scientists have sought a way to boost one of the body's most powerful natural antioxidant enzymes, superoxide dismutase, present both inside and outside of the cell membranes. The superoxide dismutase is one of the body's primary internal antioxidant defenses, and it plays a critical role in reducing the oxidative stress implicated in atherosclerosis and other life-threatening diseases. Studies have shown superoxide dismutase can play a critical role in reducing internal inflammation and lessening pain that's associated with conditions like arthritis. Until recently, attempts to supplement with oral preparations of pure superoxide dismutase enzyme proved disappointing, since the superoxide dismutase protein molecule is easily deactivated by the harsh acids and the enzymes contained in the digestive tract. Scientists have conquered these challenges by creating bioavailable forms of superoxide dismutase using natural plant extracts. When delicate superoxide dismutase molecules are coupled with a protective protein derived from wheat and other plants, they can be delivered intact to the intestines and absorbed into the bloodstream, thus effectively enhancing the body's own primary defense system. Once in circulation in the bloodstream, these powerful antioxidants go to work detoxifying potentially harmful substances and reducing oxidative stress that might otherwise contribute to aging and crippling diseases like atherosclerosis, stroke, and arthritis. By strengthening the body's primary antioxidant system, novel superoxide dismutase boosting supplements can offer the most powerful free radical protection available today. The antioxidant enzymes produced within our bodies are complex proteins that often incorporate minerals like selenium or zinc into their intricate structures. These antioxidant enzymes serve as the body's most potent defense against free radicals and the ensuing inflammatory reactions. They include glutathione peroxidase, catalase, and perhaps the most important internally generated antioxidant of all, superoxide dismutase. In the liver, enzymes like glutathione peroxidase act as catalysts, facilitating reactions that render the toxins less harmful. An enzyme known as superoxide dismutase is one of the body's most powerful antioxidants. Although there's ways to boost the superoxide dismutase production, until recently there's not been an effective oral supplement. All this changed with the development of a superoxide dismutase wheat gliadin formulation that resists the degradation by stomach enzymes and reaches the bloodstream intact. Some of the most potentially harmful substances in the body are not toxins that enter from the external environment, but rather internally generated pro-oxidants. While oxygen is essential for life, its use comes at a cost, as it poses a potential threat to biological systems. Accordingly, living systems require an entire system of compounds dedicated to neutralizing oxygen's harmful effects. Specialized functional compartments within our cells utilize oxygen in combination with other molecules to generate the energy that powers many biological processes. Without oxygen, we couldn't exist. However, in the process of generating energy by burning nutrients with the oxygen, certain rogue oxygen molecules are created as inevitable byproducts. Known as free radicals and reactive oxygen species, these unstable, highly reactive molecules play a role in cell signaling and other beneficial processes when they exist in benign concentrations. But when their numbers climb, as can occur as a result of aging and other conditions, they can wreak havoc with other molecules with which they come in contact, like DNA, proteins, and lipids, and as such, these pro-oxidant molecules become especially toxic. In fact, a prevailing theory of disease and aging states that the gradual accumulation of pro-oxidant molecules and the harm that they incur is responsible for many of the adverse changes that eventually causes various diseases. These include cancer, probably triggered by free radical-induced damage to the cellular DNA, and inflammatory and degenerative diseases like Alzheimer's, arthritis, atherosclerosis, and diabetes. While scientists have not yet reached a consensus on the topic, accumulated evidence overwhelmingly identifies increased oxidative stress with age as a source of damage to cellular structure and function. As another example, consider the visible effects of free radical damage to collagen, which forms the skin's scaffolding. Healthy collagen is responsible for the skin's elasticity and to no small degree its youthful appearance. As we age, internally generated reactive oxygen species gradually damage the molecular structure of collagen, eventually producing outward signs of aging, like skin wrinkling and sagging. For the first time, scientists at the University of Aarhus in Denmark have shown that SOD plays an important role in preventing this damage from occurring. The Danish researchers discovered that the SOD binds directly to the collagen, where it protects it from oxidation. Reports in the Journal of Biological Chemistry have noted the superoxide dismutase significantly protects type 1 collagen from oxidative breakdown, and furthermore they noted this interaction can play an essential physiological role in preventing fragmentation of collagen during oxidative stress. Superoxide dismutase is arguably the body's most crucial antioxidant, and is responsible for disarming the most dangerous free radical of all, the highly reactive superoxide radicals. Superoxide radicals, or anions, negatively charged atoms, are produced when oxygen gains an excess electron, and this occurs through normal metabolic processes, such as the catalytic transformation of various molecules by enzymes. Superoxide dismutase is responsible for catalyzing the conversion of superoxide to elemental oxygen and hydrogen peroxide, and this transformation is called dismutation, hence the enzyme's name. Although hydrogen peroxide is also a pro-oxidant compound, it's subsequently converted by the enzymes catalase and glutathione peroxidase to simple water and oxygen. Although the superoxide dismutase benefits go beyond the mere neutralization of the superoxide anions, the threat of exposure to the superoxide should not be underestimated. Superoxide anions are strongly implicated in the development of numerous degenerative diseases like atherosclerosis, stroke, heart attack, chronic and acute inflammatory conditions, and various other age-related disorders. For example, scientists at the University of Pittsburgh note the overproduction of reactive oxygen species is associated with the development of conditions ranging from cardiovascular disease to neurological disorders and lung pathologies. According to these scientists, superoxide dismutase is an ideal candidate for preventing the cell and the tissue damage that's initiated by the reactive oxygen species such as superoxide. A Texas neuroscientist noted that chronic pain associated with inflammation appears to be mediated by superoxide. Conversely, experiments have shown that pain is decreased when superoxide is neutralized. Arthritis is another condition where superoxide is implicated. Korean researchers have demonstrated the superoxide dismutase and glutathione peroxidase are significantly less active in rheumatoid arthritis patients than in normal control subjects. Not surprisingly, dietary intake of antioxidants was also lower among arthritis patients than among the controls. Superoxide also wreaks havoc by reacting with nitric oxide to form peroxynitrate, another highly reactive molecule that subsequently induces cellular and tissue injury. Peroxynitrate is implicated in several diseases like stroke, Alzheimer's, and atherosclerosis. Americans spend billions of dollars each year on pharmaceuticals designed to lower cholesterol and thus avert atherosclerosis. However, one research team recently pointed out that low SOD levels can play a greater role than elevated cholesterol in the development of this life-threatening condition. According to the researchers, diminished levels of SOD and total antioxidant status may well play a larger role in the development of atherosclerosis than isolated elevations in total cholesterol or triglyceride levels. Given the connection between superoxide and illness, scientists have long sought ways to boost the SOD levels in order to combat the oxidative damage and to reduce the incidence or the severity of disease. As one research team noted in a recent journal article, the SOD may be an effective antioxidant therapy for managing the detrimental consequences of inflammatory diseases, as well as for mitigating other conditions associated with the uncontrolled overproduction of superoxide. In the 80s, scientists attempted to treat osteoarthritis by injecting SOD derived from the blood cells of livestock directly into diseased joints. Relief from inflammation was often dramatic in these early experiments, as the injected SOD scavenged and neutralized the pro-inflammatory superoxide anions. This technique was far from practical, however, and it was never embraced as a viable treatment for human patients. Efforts to boost the SOD levels didn't stop there. However, turning to nature, scientists discovered that SOD and other primary antioxidants like glutathione peroxidase and catalase are produced by certain plants, including the sprouting seeds of crops like wheat, corn, and soy. These young grains harbour an abundance of powerful antioxidants, which can serve to protect the fledgling plant from various environmental insults. Melons also manufacture some of these antioxidant proteins, and the fruit with the highest concentration of these beneficial enzymes have significantly longer shelf life. Some plants produce SOD naturally. However, when SOD is ingested in the body, it's quickly destroyed by stomach acid and the intestinal enzymes, and virtually no SOD enters the bloodstream. Fortunately, it's possible to boost the levels of this antioxidant by consuming supplements that supply concentrated amounts of appropriate precursor molecules. Wheat sprouts represent one rich source of the SOD boosting building blocks, and it's been shown to significantly increase the internal antioxidant levels. Scientists have proposed elevated levels of one form of glutathione, the enzyme glutathione reductase, can serve as a predictor of longevity. Falling levels of glutathione are associated with diseases like AIDS, respiratory disease and infection, osteoarthritis, Alzheimer's, and even aging itself. Conversely, increased levels of glutathione are associated with improvements in these conditions. Wheat sprout enzymes are another source of bioactive plant flavonoids, and their potential benefits range from improving symptoms of fibromyalgia and joint pain to increasing energy and relieving the symptoms of chronic fatigue. These benefits are likely related to the presence of several potent natural antioxidant enzymes, including superoxide dismutase, glutathione peroxidase, and catalase. Consuming wheat sprout extract is an excellent way to increase one's level of natural antioxidant enzymes. The sprouting process promotes increased enzyme activity, and this makes the grain sprouts ideal for human enzyme enhancement. Italian researchers recently published an analysis of the antioxidant content of wheat sprout extract, noting that catalase and peroxidase activity appear very strong. They also reported that biologically active substances from the wheat sprout can be at least partially absorbed during the digestion process. The Italian scientists compared the antioxidant activity of wheat sprout extract to known pure antioxidants like ascorbic acid, quercetin, and reduced glutathione, and they found the oxygen superoxide scavenging activity demonstrated by the wheat sprout is comparable to that of pure antioxidant compounds. Research has also demonstrated that sprout enzymes possess powerful anti-mutagenic properties. That is, they help to prevent mutations that can lead to the development of cancer. According to unpublished research compiled by scientists at the University of Hawaii, in a survey of 120 subjects who ingested large amounts of plant-based antioxidant enzymes, 88% reported increased energy and 72% reported feeling stronger. 82% of survey responders reported feeling better overall after supplementing with sprout-derived antioxidants. SOD is a large molecule that when orally consumed is not readily available by the body. However, technological advances have enabled scientists to bond the SOD, the extract from a type of cantaloupe melon that naturally produces high levels of the enzyme, to a biopolymer extracted from ordinary wheat. Studies have shown that the wheat component, known as gliadin, protects the fragile SOD molecule from the ravages of the stomach acid and the intestinal enzymes, thus allowing the molecule to enter the bloodstream intact. The success of this novel pairing has been demonstrated in animal and human studies. Experiments in France show that the SOD gliadin, but not the SOD alone, caused a significant increase in circulating antioxidant enzyme activity when consumed by the lab rodents. This increase in the SOD was associated with an increased resistance of the red blood cells to oxidative stress-induced rupture, according to the research. In another experiment, the French team examined the antioxidant and anti-inflammatory properties of SOD extracted from melon. In both lab cell studies and live animals, their studies showed that the antioxidant properties attributed to the melon extract were indeed due to the active SOD. The SOD prompted immune cells or macrophages to release the anti-inflammatory cytokine interleukin-10 rather than inflammatory tumor necrosis factor, which the cells can release under conditions of oxidative stress. Subsequent studies of live animals showed the SOD levels increased when the SOD gliadin was administered orally. This led the scientists to conclude that it's possible to elicit the pharmacological effects of the antioxidant enzyme in the animal subjects. More recently, Japanese scientists studied the effects of oral SOD gliadin on tumor development in lab rodents. Gliadin alone, or the SOD gliadin combination, was administered orally to the test animals before and during the experimental tumor induction. About 80% of gliadin-only subjects developed tumors, but only half as many animals with the SOD gliadin group did. Furthermore, tumor cells from the group that didn't receive the SOD gliadin supplement exhibited signs that they would readily spread or metastasize. By contrast, tumor cells from animals in the SOD gliadin group showed decreased metastatic ability. In the reports published in the British Journal of Cancer, the Japanese researchers noted that orally active SOD prevented tumor progression promoted by inflammation, and that this might have elicited these effects by scavenging the inflammatory superoxide anion. To test the hypothesis that the SOD gliadin improves antioxidant defenses in humans as well as animals, German scientists subjected 20 volunteers to an hour of hyperbaric oxygen treatment. During the procedure, 100% of the oxygen was forced into the bloodstream at 2.5 times the normal atmospheric pressure. Hyperbaric oxygen treatment, though medically necessary on occasion, is known to induce oxidative stress. This stress may be quantified by measuring breaks that occur in strands of the DNA and by monitoring levels of isoprostanes. These indicate the oxidative damage to the cellular membranes. Hyperbaric oxygen treatment is also known to reduce levels of antioxidant enzymes such as red blood cell SOD and catalase. In a randomized, double-blind, placebo-controlled study, scientists demonstrated that supplementing with the SOD gliadin significantly decreased the oxidative damage resulting from the hyperbaric oxygen treatment. The investigators found that an orally effective SOD wheat gliadin mixture can protect against DNA damage while also preventing elevations in isoprostane levels. These findings suggest that the SOD can therefore be considered a powerful antioxidant. A previous experiment by another research team found hyperbaric oxygen treatment induces breaks in the DNA strand. In an effort to reduce this oxidative damage, the scientists administered oral antioxidants like vitamin E or N-acetylcysteine prior to the treatment, but these measures failed to protect against the oxidative damage induced by the hyperbaric oxygen treatment. This discrepancy would seem to indicate that effective protection against oxidative stress requires increased levels of the primary antioxidant SOD, as opposed to a boost in a secondary antioxidant like glutathione. Primary antioxidants like superoxide dismutase are our first and most important line of defense against highly reactive, potentially destructive, oxygen-derived free radicals. Researchers believe SOD decreases with age, and evidence suggests that boosting the falling SOD can help to guard against diseases and extend lifespan. In the past, it was difficult to raise levels of these beneficial enzymes. However, it's now possible to bolster a weakened antioxidant defense with nutritional supplements that include orally bioavailable SOD and other primary antioxidants. For more information about herbs, supplements, and natural treatment plans, check out my website.